We know that antibodies play a major role in protection of our body against harmful pathogens and toxins. For our convenience, we can divide functions of antibodies into natural role of antibodies and artificial role of antibodies. Among natural role or functions of antibodies, it carries out two principal functions in immune defense. The first function is to recognize and bind to foreign material or antigen, and the second function is to trigger elimination of foreign material or antigen. Recognition and binding to foreign material generally means binding to molecular structures on the surface of the foreign material, also known as antigenic determinants, that differ from molecular structures made by the cells of the host. These antigenic determinants are also called as antigenic epitopes. These are molecular patterns that are usually expressed in multiple copies on the foreign material, such as proteins or carbohydrates on a bacterial cell surface, or envelope spikes on the surface of a virus. Antibodies of a single host can recognize a huge variety of different molecular structures, and a human is capable of producing antibodies against billions of different molecular structures. The simple act of antibody binding may be sufficient to inactivate a pathogen or render a toxin harmless. For instance, antibody coating of a virus can prevent entry into target cells and thereby neutralize the virus. In many instances, a second function of the antibody molecule is deployed to trigger the elimination of foreign material. In molecular terms, this involves the binding of certain molecules called effector molecules to antibody-coated foreign material to trigger complex elimination mechanisms, such as the complement system of proteins, phagocytosis by host immune cells, for example, neutrophils and macrophages, and antibody-dependent cellular cytotoxicity, ADCC, by natural killer cells. Let's discuss all of these functions one by one, starting with antibodies and complement. Among different classes of antibody, IgG and IgM play role in complement activation. IgG molecule cluster together on the surface of pathogen, such as a bacterium, by its FeA-BU region. This complex leads to the binding of complement C1 molecule via the hexavalent C1 subcomponent. This attachment or binding is done at Fc region of IgG. It triggers the proteolytic activity of the C1 complex, and thus activates classical complement pathway. That ultimately leads to the formation of C3 convertase. That can cleave C3 into C3a and C3b. The resulting C3b is then converted to C5 convertase, which generates C5a with chemotactic and anaphylactic functions, and C5b, which forms the first component of the membrane attack complex. Other than IgG, IgM can also aid in complement activation by following slightly different mechanism. It is already clustered, pentameric, but occurs in an inactive form. Binding to multivalent antigen appears to alter the conformation of the IgM molecule to expose binding sites that allow C1 to bind and the classical pathway of complement to be triggered. So in this way, antibodies play an important role in complement activation. Another role antibodies play is in phagocytosis. Antibody and phagocytosis. We know that phagocytosis is the process of engulfment of microbes inside the phagocytic cells, mainly macrophages, neutrophils, and dendritic cells. It is an efficient process for microbial killing and digestion. But sometimes microorganisms become able to resist phagocytosis, and thus they need proper markers for their recognition and proper engulfment by phagocytosis. So here antibodies play their role. Antibodies, mainly IgG, binds with the antigen via FeA-B segment. In other words, these antibodies coat the antigen so that it can be engulfed. This coating is done by a process called opsonization, and the molecule that covers the antigen are called opsonins. So here antibody acts as opsonin. Phagocytic cells have specialized Fc receptors. Through these Fc receptors, they binds to the Fc region of antibodies surrounding the antigen. For opsonization, 2 or more antibodies bound to the microbe. A single molecule complex to microbe is not enough because it cannot cause the cross-linking of the Fc receptors on the phagocyte surface membrane that is required to activate the cell. In this way, antibodies play a role in phagocytosis as well. Another function of antibody is in antibody-mediated cellular cytotoxicity. It is also known as antibody-dependent cell-mediated cytotoxicity, or ADCC. It is a mechanism by which antibody-coated target cells are destroyed by natural killer cells or cells with low affinity Fc receptors like monocytes and neutrophils. But mainly this mechanism involves natural killer cells through the CD16 receptor that binds the Fc portion of IgG antibodies, triggering the lysis of targeted cells. The overall importance of ADCC in host defense is unclear, but it represents an additional mechanism by which bacteria and viruses can be eliminated. So these were the natural functions of antibodies. But antibodies play some roles artificially as well, including antibody role in passive immunity and role of antibodies in therapeutics. Antibodies can provide passive immunity to the individuals who needs additional support for immune defense against pathogens, or those that needs quick immune protection. We know that once antigen enter the body, antibodies are produced against them. But in those individuals that have compromised immune system, or due to any other reason they are unable to produce antibodies in required amount, then antibodies from outside source are directly introduced in their bloodstream. It provides efficient and quick protection against that invading antigen. Antibodies provide passive immunity naturally as well, when they travel from mother to the child during breastfeeding and provide protection to the baby until his own immune system gets fully prepared to fight against infection. Another artificial role of antibodies is in therapeutics. Antibodies and fragments of antibodies are also used in the field of therapeutics, where they can target disease-related molecules. Therapeutic antibody recognizes and binds to the antigen receptor to activate or inhibit a series of biological processes for blocking cancer cell growth or triggering immune system. Because of their high affinity and specificity, their ability to target diverse molecules, and their ability to be modified and refined by genetic engineering techniques. In therapeutics, antibodies can be used as immunosuppressive agents, like during organ transplant, so to avoid rejection, anti-cancer agents, where they suppress the uncontrolled division of cancerous cells. Also, they play a role in cardiovascular diseases, respiratory diseases, infection, ophthalmologic disease, and so on. So this was all about the functions of antibodies. This video must have improved your knowledge about the structure and functions of antibodies. For more such videos, keep watching sqadia.com.